I'm really happy to be here and present my research work on microbiota characterization through the digestive tract of horses fed a high fiber versus a high starch diet. This work wanted to underline the strong relationship between the microbiota and the gut health. I focused a lot of my research activity on the study of the gut health. I'm Federica Raspa, I'm a veterinarian, and I complete my PhD in equine science with a particular focus on equine nutrition and welfare. I'm actually a researcher at the Department of Veterinary Sciences of the University of Turin within the animal nutrition and feeding sector. As I told before, I focus a lot of my research activity on the study of the effect of two different feeding management, one based on high amount of fiber versus one based on high amount of starch on gut health. And accordingly, I studied the gastrointestinal environments in terms of volatile fatty acids production, the features of the gastrointestinal wall in terms of intestinal permeability and histomorphometry features, the microbiota of course, but also, for example, how the behavioral activity changes in relation to the diet. Before to describe more about the effect of the diet on the microbiota, I think that could be useful to remember what is gut health, because the concept of gut health is becoming progressively more important in the field of animal nutrition, and it is often considered a synonymous of animal health. In human medicine, gut health is often defined as absence of clinical disease, but you can understand that this definition is not suitable when we talk about animals. Generally, in veterinary medicine, there is not a consensus about the definition of gut health, but some authors have proposed this sentence, so a multidimensional concept that depends on the maintenance of a delicate balance between the host, the gut microbiota, the structure and the function of the intestinal barrier and the dietary compound. Of course, when we talk about gut health, we should remember that we need to respect the food pyramid of the horse. So this food pyramid is characterized by the forage as the basis. The forage, because we should remember that horses are grazing herbivorous, monogastric and in-gut fermenters. So as herbivorous, forage should be the first dietary compound that we that we use when we plan its diet. So forage is the basis, and when we talk about forage, we talk about of grass when pasture is available, straw, for example, but also hay and the silage. Just to give you some number, how much forage should be fed. The literature told us that horses should be fed at least 1.5% of their body weight in dry matter per day. That means that 500 kilogram horses should be fed at least 8.5 kilogram of hay if this hay is around 90% of dry matter. So it is needed to perform a calculation. Remember that grass, silage, and hay have different dry matter content, so be sure that the quantity of hay, of forage, is adequate on the basis of the forage that you offer to the horse. Continuing to describe the food pyramid of the horse, we often have to supply also concentrate feed. Concentrate feed are needed to increase the energy content of our ration, but it is not always needed. When a concentrate feedstuff is supplied, we should take into account some rule so that horses should be not fed more than 1 gram of starch kilogram body weight meal and not more than 2 grams of starch kilogram body weight day. This means that a 500 kilogram horse can digest not more than 500 grams of starch meal, thus 1 kilogram of a cereal-based concentrate. These are safe level of starch, so it is really important to remember this rule. Obviously, the food pyramid of horses is characterized by the top of the pyramid that is always important. So the integration of the diet, the supplementation with mineral and vitamin and amino acids is important to adequately balance the diet in terms of mineral and protein content. Why horses should be fed with a safe level of starch, that is that we had described in the previous slide. There is a physiological reason, because the horse's capacity to digest starch is limited, since they have a low production of pancreatic alpha amylase. So when we feed horses with high amount of starch, what happens is that the starch is not totally digested by the alpha amylase within the small intestine, and the indigested starch reach the ingut, where it can trigger microbial population changes during the fermentation process. And in that cases, so when the diet is not balanced for forage concentrate content, what we have is a lot of effect not only on the microbiota but also on the gut environment. So the volatile fatty acids changes, the body condition also changes, but also the morphology, the integrity of the gastrointestinal barrier and the behavioral activity of the horse according to the microbiota gut-brain axis. So if forage is the basis, unbalanced diet can be in which a high amount of starch is fed to horse represent a common mistake. Above all, in some sporting discipline, for example, racing horses, is reported that are really fed high amount of starch, but this is lead to high prevalence of gastrointestinal disorders with high morbidity and high mortality rate. What we know in the scientific literature, that gut health is represented by this interplay of factors that but most studies have used fecal sample for microbiota characterization. My question is, are the faces representative of the proximal compartments. So the microbiota of the feces reflect the microbiota that we can find in the other intestinal compartment, considering that the gastrointestinal tract of the horse is really huge, and the ingut is a real fermentation chamber that represents the 40% of its body weight, so that a 500 kilogram body weight horse is characterized by 200 liters of digestive tract for a total length of more than 30 meters. What we found in the scientific literature is that a few studies have characterized the microbiota in different intestinal compartments. Some of these studies have used culture-based methodology or PCR-based methodology, but these kind of methods do not allow a full characterization of the richness and diversity of bacterial population. Only one study have used the NGS methodology to characterize the microbiota along the digestive tract of the horse, but in this study the author used a few number of horses not homogeneous for breed and age, and that were euthanized for pathologies that were unrelated to gastrointestinal disease. The aim of the present study was to apply the NGS approach in healthy horses homogeneous for breed and age, in order to understand the microbiota changes in different intestinal compartments according to feeding management. One group of horses was fed a forage-based diet, and one group of horses was fed a high-starch diet. The study was performed in one real farm, so not in one experimental farm of my region, Piedmont, in northwest of Italy. This farm is a farm that rears horses specifically for meat production, so the horses involved were destined for slaughter, and we involved 19 Bartigiano horses of different sex but homogeneous for age, because they were around 14 months. As you can see in the picture, horses were housed in group, were randomly divided into group pens, and they were housed in the same environmental conditions, so there were no difference among animals according to environment or housing system. The only difference was related to the feeding management. So one group of horses was fed a starch-rich diet according to the usual feeding management of the farm. One group of horses was fed a forage-based diet that was planned by the nutritionist according to the nutritional requirements for horses suggested by the Institut National de la Recherche Agronomique, so the French Institute INRA. So here you can see the daily nutritional composition of the diet that horses were supplied. The diet were not isoenergetic or isoproteic, but what I want to show you is the difference in the starch consumption per meal that was really high for the starch group, and respect the safe level for the forage-based group. After a period of 129 days of a different feeding system, horses were slaughtered, and at slaughter, different intestinal compartments were sampled. In particular, what we collect were the digestive content of the small intestine, so the duodenum, the jejunum, the ileum, and of the gut, so the apex of the cecum, the sternal flexure, the pelvic flexure, the right dorsal column, and the feces collected by the rectal ampulla. The digesta sample were collected in 2 ml Eppendorf and in an aseptically way, and were immediately stored at less 80 degrees until DNA extraction. So microbial diversity was studied by sequencing the amplified V3 and V4 region of the 16S rRNA gene. After sequencing, raw reads were analyzed in our environment by applying specific statistical analysis. First of all, I wanted to remember the difference between alpha and beta diversity. Alpha diversity means how rich and diverse the microbiota is within a single sample. The beta diversity describe how the microbiota varies between different samples. These concepts are for us important because allow to understand the results of our study. So, alpha diversity was measured through the Cow and Shannon indexes and the observed richness, species richness. The cow index was higher in the cecum of horses fed the high forage diet compared to the high starch diet. The Shannon index was lower in the ileum of horses fed the high forage diet, but was higher in the cecum, pelvic flexure, and right dorsal column of the horse fed the high forage diet. Moreover, the observed species richness was higher in the right dorsal column of the horses fed the high forage diet compared to the high starch diet. This represents a first important finding, because the horses fed the high forage diet show higher alpha diversity indexes in the cecum, pelvic flexure, and right dorsal column compared with horses fed high starch diet. This is a relevant finding because high levels of bacterial diversity have been associated with greater stability of the intestinal microbiota against stressors and an increased resistance against potentially pathogenic bacteria. Considering the measure of beta diversity, this relieved significant differences between the different intestinal tract compartments according to the diet. This was not surprising since the diet is well known to be the main factor affecting microbiota composition in the horse. Moreover, as you can see in the chart, the samples form two distinct clusters, differentiating between the upper and the lower digestive tract of the horse. The present heat map show that at lowest taxonomic level, cluster analysis of the most frequent amplicon sequence variants confirm the presence of more clusters. In particular, one cluster comprised the cecum and external flexures, and the other was characterized by the pelvic flexures, the right dorsal column, and the rectum. These results confirm what other authors have shown, that the neighboring intestinal tract compartments are more similar to each other. Considering the microbiota composition at phylum level, Firmicutes was the dominant phylum in both the horses fed the forage diet and starch diet, and Firmicutes phylum outnumbered Bacteroidetes, Proteobacteria, and Vermicrobia phyla. But some differences can be described at family genus level, in particular within the small intestine, and in particular within the jejunum and in the ileum. The horses fed the high starch diet display a higher relative abundance of proteobacteria compared with horses fed the high forage diet, as a result of a higher relative abundance of the Enterobacteriaceae and the sulfovibrio. Previous research published by our research team on the same horses fed the same diets show that the horses fed the high starch diet were characterized by greater levels of inflammation of the intestinal wall in the jejunum and by higher level of intestinal permeability. In fact, the increased presence of proteobacteria is reported to be associated with inflammatory intestinal disease, in fact, Enterobacteriaceae comprises some pathogenic bacteria such as Escherichia coli and Salmonella species, which can lead to dysfunction of the intestinal barrier, generating a condition of leaky gut and enteritis. The section of the intestinal wall that we found at slaughter reveal at macroscopic level a really strong inflammatory condition of the intestinal wall of the horses fed the high starch diet. Here you can see a section of the jejunum. In particular, this kind of evidence were confirmed by the histological finding that reveal an increased inflammation of the wall of the jejunum and the pelvic flexure in particular. Within the hingut, and in particular considering the cecum and the pelvic flexure that we saw to be close in terms of microbial composition when I show you before the heat map, we found an increase in metabolic pathways involved in the fermentation process with the production of short-chain fatty acids. In fact, we found a significant increase of some bacteria families, so Pybrobacter, Prevotella, and Paraprevocellaceae, Paraprevotellaceae that are involved in the core microbial bacteria of the horse and are important for fiber digestion. Within the cecum and external flexor, moreover, we have a reduction in the horses fed the starch diet of the overall size of the core microbiota, with an increase of some species like Streptococcus and Fusobacterium that are potential pathogenic bacteria associated with the presence of colitis or diarrhea. Considering the distal compartments, so the pelvic flexor, the right dorsal column, and the feces collected by the rectum, we found an increase in horses fed the forage diet of Akkermansia, a mucin-degrading genus that helps to maintain the integrity of the mucin layer of the intestinal barrier, and an increase of the family belonging to Lachnospiraceae and Ruminococcaceae in the fecal samples, which form part of the intestinal core microbiota of the horse. In conclusion, we can state that each intestinal tract compartment behave as an ecological niche that should be treated separately, and diet significantly influences the microbiota composition, and probably to understand the changes occurring at lower taxonomic level along the digestive tract, it is not enough to analyze fecal samples collected from the rectum. Our take-home message is that the feeding management has a key role to safeguard the gut health and the healthy microbiota. It is needed to respect the food pyramid of the horses, for which forage is the basis. A forage-based diet is crucial because it increases the microbiota diversity and richness. A forage-based diet increases the presence of short-chain fatty acids producing bacteria, and differences in microbiota composition are present between the small intestine and the ingut, and between the selected intestinal compartments. Fecal samples may not be useful to represent the changes occurring in the proximal compartments. I really thank you for the time dedicated to me. I'm happy to answer to your curiosity and question. Thank you so much.